Look who's laying comfortably on my makeup chair and will have to be kicked out in a few minutes. So you guys, outfit of the day is going to be extremely simple. I'm going to wear my black legging jeans from Uniqlo, and I'm going to wear this beautiful orange sweater which has this very nice detail here of the sleeves, and I'm going to pair this outfit with my Ana Luisa jewelry. I'm gonna wear the celestial element rings that I've shown you before. I'm going to wear these two rings, which you might have not seen before because they are new. And I'm going to wear this bracelet, which is not from Ana Luisa. It's from someone, but I can't remember who it's from. Of course, a watch. And for today's perfume, I have chosen my Nina Ricci Apple, which I'm trying to finish so hard. As I mentioned, you guys, the outfit is exceptionally simple, so there's really nothing much to it. Of course, these are going to be substituted for heels later on. But like I said, I really enjoy the sleeves of this shirt because they like kind of widen here at the end, and I just think that looks really cute. He wasn't too pleased, but I made him a little nest with the blanket, and I think he did settle very cutely in there. Alright you guys, today we're moving into number 4 into my ranking, and this for me is the Divine Rose 2 palette, which, as you guys know, or you may not, is the latest big mothership release from McGrath Labs. In my humble opinion, this is the best palette that has come out this year. It's a bit of a spoiler for my palette ranking of the year, but I don't think this palette has a contender that can stand up against it. It's just nothing can compare to the color story, the vibrancy, the variety of textures, the versatility of this palette, and just how much fun it is. I adore Divine Rose 2. I'm actually going to be doing my classic look with this palette, which would be this in my crease, these two on my lids, with this one towards the center as well as in the inner corners, and I might actually apply a little bit of this shade to deepen up. I usually don't do that in the summer, but I might do it today. This is a relatively simple look, but it is definitely my favorite one to do with this palette. Besides, of course, using the multi-chrome shade all over my lid and swooning every time I stand in front of a mirror. So for today's story time, I was thinking what to tell you, and I thought maybe I can try telling you a little bit about my job. And today I'm going to start out with my early days as a scientist towards the beginning of my PhD, and actually a discovery I made during my master's internship, which became the basis of my PhD project. Now, please feel free to tell me in the comment section below if you find this extremely boring and you don't want me to talk about this, because I will definitely respect your wishes in that regard. But if you guys found it interesting, then next time I might tell you about what I'm doing currently as a postdoc, and you know, we can talk a little bit about what I actually do throughout most of my day. So starting off the look with the peachy matte shade Naked Rose, I think it's called, or Naked Blush through my crease. I came to the Netherlands to do a master's in 2006, and then my first internship was in a hematology lab, a lab which studies the basic biology of hematological cancers. The reason I chose this lab and the reason I was fascinated with hematology is because when I was 11, I was admitted to a hospital with to this day unresolved iron deficiency anemia, maybe because I was growing too fast or I had just gotten my period. For whatever reason, around the age of 11, I had a really severe drop in my iron levels, and I had to be admitted to the hospital for about a week to receive blood transfusions. But during my week in that hematology ward, it was like a pediatric hematology ward, there were kids who were much sicker than me, and I could, like a part of me, even though I was a kid, could tell that these kids are much sicker than I was. I think these kids had cancer. And I think that kind of left an impression on me, and like this desire to do something for them. So when I had the chance to choose an internship in hematology, I immediately jumped onto it. Now, it was not pediatric hematology, it was actually more oriented towards a disease which is not even cancer. My supervisor was at the time a PhD student who was actually a hematologist in training. Like, she had an MD and she had taken some time off to do a PhD, because especially in hematological cancers there is so much known about the molecular defects. A lot of MDs who pursue hematology actually do a PhD in like the basic biology of hematological cancers, blood cancers if you will. So my supervisor at the time was studying a disease called myelodysplastic syndromes. And myelodysplastic syndromes is this very mysterious condition which for the longest time was actually considered to be like a pre-leukemia stage. So a large percentage of the patients do eventually develop leukemia, but a lot of them just have like this indolent hematological disorder where essentially what it means is that their blood cells, their precursor blood cells, don't develop fully into the mature blood cells, and they remain in like various stages of their differentiation, so their like maturation, and they just look super weird under the microscope. And because of that, these patients have of course a lot of problems with their blood cell counts, with infections. So what my supervisor at the time was doing, she was trying to find out what kind of mutations occur in these patients, because certain mutations would mean that you can approach treating the disease in a certain way, or you can discover something new which might lead you or point you to a new type of treatment. So she had done the so-called SNP arrays, and SNP arrays stands for single nucleotide polymorphism arrays, which is basically a very fancy way to say that you have this technique where you can zoom in on the genome and find very small deletions and insertions, because it's all about resolution, you guys. The technology in that respect has gone from you can only see like an extra chromosome or an extra half a chromosome into now we can read into like single nucleotide changes into the DNA. But I think I will explain in a little bit more about the DNA. So she was looking into these types of like small deletions and insertions into the genome, trying to locate new places in the genome which are associated with myelodysplastic syndromes. And the purpose of my internship was that I was to pick up those little regions and sequence the genes which are located there to find out if they have mutations. That brings us to, let me explain what exactly all of that means. So your genome is basically a stretch about a 2 meter long, so each of your cells contains a 2 meter stretch of DNA which is tightly compacted into chromosomes. And on your DNA you have a variety of things, but the most important stuff is those little like stretches which contain sentences, which consist of 4 letters, A, T, C, and G. And these letters have a start, they have a stop, and they mean a protein. So these little sentences on your genome are responsible for encoding a protein, and the proteins are the working horses in your cell. They make things happen in the cell. Let's just put it that way. And when something is wrong with your proteins, because there is a mistake in that sentence, you get proteins which are either not there, or because of that mistake, they have a completely different function. So I was looking for those single nucleotide mistakes. So a single change, like a single letter in that sentence may change and completely change the sentence and the protein. So I was looking for those with a technique that you might have heard a lot about lately because it's been used to diagnose people for COVID. It's called PCR, which basically stands for polymerase chain reaction. And I can explain it to you because it's actually quite simple. It's basically the following. When you have your cell and the DNA in there and you want to read these sentences, it's very hard to read them if you have like only a few copies. You want to have a lot of copies of the same thing in order to be able to read through it properly. So what PCR does is basically it amplifies the same region of DNA, because you can design little stretches that fit onto the part of DNA that you're interested in. Then you can add certain reagents to it, including the enzyme, and then it will just read through the whole sentence and just make super many copies of it. Like it's an exponential rise in the copies. That is essentially what PCR does. It's just an exponential increase in a stretch of DNA. So that's what I was doing. So for months and months I was doing fruitless PCRs, testing region after region and not finding a mutation in any of them. And then finally around June, I started my internship in December, and around June I had finally reached the point where I was going to sequence the last region that was a part of my internship, and it contained a gene which at the time was a hypothetical gene, meaning that we know it's there, we have no idea what it does or anything about it. And of course we had saved that hypothetical gene for the last, because Nobody wants to focus on shit we don't know anything about. There were far more interesting targets in the initial PCRs that I did in other regions that seemed much more promising than that last region that I was going to focus on. And that last region was the 4q24, meaning the long arm of chromosome 4 and the region 24. And we had a small, I think it was a small deletion on one allele, and we were going to see whether the other allele has a mutation. And it was a very, very long gene. It is 6,000 base pairs, which is a lot. So I designed my PCRs, I started doing the PCRs, everything worked fine, and I was moving into my patient material. And then I want to say, almost at the end of my internship, I was looking through the sequences that came back from the patient DNA. And all of a sudden, I see this little nucleotide change. So, you know, I told you that the little sequences, the little stretches of DNA that signify a protein have a start and a stop. And what happened in the case of my patient was that the stop came much prematurely. So instead of at the end of the 6,000 base pairs, I think it came all the way down to the 500 base pairs. And I stared at it for a little bit, and then the first thing I thought was, I need to go to my supervisor and show her, because I think I might be hallucinating at this point. I think I am so keen on finding something that I think my mind might be playing tricks on me. So I ran to her and I was like, look, I see this, do you see the same? And then she looked through the sequence, and we were both completely exhilarated because she saw the same thing. She saw the same premature stop in our sentence. And we were both, I cannot even begin to explain to you what a high it is, because these kind of highs happen very, very rarely in science. You don't really experience that kind of adrenaline surge when you do science often. 90% of the time shit fails, or it's a negative result, so it's not very exciting. But in that, 1%, when the stars are aligned and you find something, it is the most exhilarating. It's probably like doing crack. I've never done crack, but I would imagine that that's what happens. I'm going to apply a little bit of that topper shade, pretty much over top of everything, because why not? So after months of fruitless PCRs, I finally had found something. Now, we had to confirm, of course, that what we found is not something that happens more often, so we had to also sequence a bunch of controls. So as the last part of my internship, I sequenced like 20 controls, and none of them had that mutation. It would have also been very strange, because what happens that if you have that sentence and you have a stop that comes very prematurely, that is almost always a deleterious mutation. And you can also have little switches in the letters of the DNA, which lead to a change in the code. So you don't have a stop, but you have a change in the protein, and that may or may not be deleterious. So that would have been a little bit more ambiguous, but in the case of that premature stop, it was almost 100% sure that this is a true mutation. So after sequencing those 20 controls, unfortunately I had to leave because it was the end of my internship and I had to still do another internship and finish my master's. But luckily, in the meantime, they continued doing research on this gene, and they actually found out that it is mutated in 25% of the patients with MDS, with myelodysplastic syndromes, which is really remarkable. So we made a huge discovery. It was like a really big deal. I wasn't even allowed to say the name of the gene when I was doing my final presentations for my fellow master students from the same master program. I had to substitute it with gene X. It was such a big deal. And like I said, I had to leave the lab, but my boss at the time and I agreed that we're going to write up a project for a PhD so that I can come back to the lab and study the function of this protein. What I didn't realize at the time, and what I think is really, really cool, and to this day is something that it remains like such a special experience for me, I was probably the first person in the world to hold in my hands the information about this mutation. I'm basically mirroring what I did on my upper lid. So it's a very special memory to have, and a very unusual one. Most people, like most students who do internships, don't really end up discovering very important things. I think I was just, lucky. It was serendipity, and it just worked out. It's a very special memory for me, and it served as the basis for my PhD project. Just to finish off the look, I'm going to take this purple pencil liner from Neve Cosmetics and just run it on my waterline. So you guys, this is the final look with the Divine Rose 2 palette. I always love how this look turns out. It was, you could see how effortless everything was. Everything just blended out, everything applied so beautifully, and I don't know about you, but I think there's probably nothing more stunning than this gold shade right here. I know Pat has done a lot of golds, but I think this one probably takes the cake. I have fallen head over heels in this palette, and it is definitely not something that I expected to happen, because rosy tones and like the whole rose theme hasn't really been my speed, but because I love Pat McGrath and I know that I'm probably not going to be disappointed by her products, I have bought both the Divine Rose 1 and the Divine Rose 2 palettes, and I have to say both of them have surpassed my expectations. This one especially is just something else. Now on my lips I am wearing a lipstick and a gloss. So for my lipstick I am wearing the gorgeous Velvet Dragon by Lisa Eldridge. It is this stunning yellow toned red. And over top of that, just to give it a little bit more shine and sparkle and creaminess, I applied this lip gloss from Pat McGrath, which is called Carnal Desire, which has the most beautiful like pink, red and gold sparkles to it. And I just dab a little bit of that in the center of my lips. I kind of work it into the lipstick. I don't just swipe on. I apply on the back of my hand and then I work it in with my fingers, because I feel like that way it just blends into the formula of the lipstick, and it also doesn't give me a lot of problems when I'm wearing my face mask at work. Let's wrap up this video, you guys. Let me know if you found the science too boring. Should I just cut it out and tell you other things in my story time, or would you like to hear more about my current work as a postdoc? I would be more than happy to do either of your bidding, so just let me know what your wishes are. We're going to see each other next time for palette number 3. No, well, not palette number 3, but number 3 in my ranking, which is the stunning Iconic Illumination Quad from last year's holiday collection. Thank you so much for watching. Don't forget to like and subscribe if you would like to see more of me, and I'll see you in my next video. Bye!